This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to Paternal Exercise, a Transgenerational Legacy. For generations, we've thought inheritance was all about DNA. But what if a father's lifestyle, specifically his exercise habits, could profoundly shape his children's health and endurance even before conception? This narration dives into groundbreaking scientific discoveries, revealing how sperm microRNAs carry this surprising legacy. Prepare to challenge your understanding of heredity and discover how a father's fitness can echo through generations, redefining well-being for the future. Chapter 1. Paternal Exercise, a Transgenerational Legacy. For generations, our understanding of inheritance has largely centered on the blueprint of DNA, the genetic code passed down from parent to child. We've learned about genes dictating everything from eye color to predispositions for certain health conditions. But what if the story of inheritance is far richer, far more dynamic, and deeply intertwined with the lived experiences of a parent, even before conception? What if a father's physical activity, his commitment to exercise, could leave a tangible, beneficial mark on his offspring, shaping their endurance and metabolic health in profound ways? This is not a hypothetical question, but a groundbreaking area of scientific discovery. Recent groundbreaking research, notably a pivotal study published in Cell Metabolism in October 2025 by a collaborative team led by Xin Yin, Chenyu Zhang, Shi Chen, and Tao Zhang, is revealing a truly surprising legacy, one that challenges our conventional notions of heredity and opens up new pathways for understanding health across generations. This isn't a purely academic pursuit. It stems from observations, such as lead investigator Xin Yin's undergraduate curiosity about why children of athletes often seem to possess a natural talent for sports. This cutting-edge biological research offers insights that could redefine preventive medicine and personal well-being for years to come, captivating not only scientists, but anyone interested in the intricate dance between lifestyle, biology, and the future of human health. Throughout the vast tapestry of evolutionary history, the capacity for sustained physical activity was not merely a desirable trait, but a fundamental prerequisite for survival. Our ancient ancestors, whether hunting for sustenance, migrating across vast landscapes, or evading formidable predators, relied intrinsically on their physical prowess. Their very existence and the propagation of their lineage hinged on robust endurance and efficient metabolic function. Fast forward to our modern world, and the landscape has dramatically shifted. Sedentary behaviors and physical inactivity have, for many, become the norm. The immediate life-or-death imperative for intense physical exertion has largely receded, making exercise seem, at times, less essential for day-to-day survival. Yet a burgeoning body of scientific inquiry is unequivocally demonstrating that the biological benefits of exercise transcend mere individual fitness. These advantages reach deeper, extending their influence to the physiological and metabolic characteristics of future generations. While a significant portion of earlier research understandably focused on the profound impact of maternal health and exercise during pregnancy on offspring development, a captivating new spotlight is now illuminating the often underestimated role of the father. It turns out that fathers may transmit a wealth of epigenetic information to their offspring, not just through their DNA, but through other remarkable molecular carriers within their sperm. This discovery expands our understanding of inheritance beyond the genetic code, delving into the realm of how environmental exposures and lifestyle choices can leave an indelible non-genetic imprint on the next generation. The core of this groundbreaking revelation lies in the observation that offspring sired by fathers who engaged in regular exercise exhibit remarkable intrinsic adaptations to physical exertion and possess improved metabolic parameters, even without having exercised themselves. Imagine children born with a natural head start in endurance and metabolic efficiency simply because their father maintained an active lifestyle before their conception. This is the surprising legacy researchers are uncovering. To unravel this intricate connection, scientists conducted carefully designed experiments, often utilizing animal models like mice, which provide invaluable insights into fundamental biological processes. In one such study, male mice were divided into two groups. One underwent a structured 8-week endurance training program on a treadmill, steadily increasing their running speed and duration, transforming them into veritable mouse athletes. The other group remained sedentary, living in standard cages with unrestricted movement, representing a more inactive lifestyle. The differences in the fathers themselves were striking. The exercise-trained fathers exhibited lower body weights, significantly increased lean muscle mass, and reduced fat mass. Their bones were denser, indicating improved skeletal health. When put to the test on the treadmill, these fit fathers ran considerably longer distances and for extended durations before reaching exhaustion, a clear testament to their enhanced endurance capacity. To ensure these adaptations weren't at the cost of mental or physical impairment, researchers observed their general locomotor activity and anxiety levels, finding them to be normal. Furthermore, indirect calorimetry, a method used to measure energy expenditure, revealed that these exercised fathers had significantly higher oxygen consumption and overall energy expenditure compared to their sedentary counterparts, indicating a more efficient and active metabolism. The truly captivating part of the research began when these exercise-trained and sedentary fathers were bred with female mice. The question was, would the physical and metabolic advantages acquired by the fathers be passed on to their offspring? The results were unequivocal. The adult offspring of the exercise-trained fathers, even though they themselves had never undergone any training, displayed a remarkable inheritance of their father's endurance capacity and improved metabolic health. These F1 generation offspring of active fathers showed no abnormal growth or weight gain, developing normally. However, when their physical capabilities were assessed, they displayed significantly enhanced endurance and improved physical resilience, outperforming offspring of sedentary fathers in various tests of sustained effort. Beyond physical performance, the metabolic health of these offspring was also significantly enhanced. They exhibited a more favorable body composition and a more robust, efficient metabolic profile, indicative of intrinsic biological adaptations rather than simply increased movement. Explain with examples, but do not use analogies that are not truthful. These compelling observations from animal models provide robust evidence that a father's exercise habits can indeed leave an enduring, beneficial legacy on his children's physical capabilities and metabolic well-being. But how exactly does this legacy get transmitted? It's clear it's not through direct genetic inheritance of the father's specific muscle traits, as the offspring themselves didn't exercise. This points to a fascinating mechanism known as epigenetics, changes that influence gene activity without altering the underlying DNA sequence. The emerging understanding is that tiny RNA molecules, particularly microRNAs, found within sperm, act as crucial messengers. These molecular couriers are reshaped by the father's exercise, carrying information that can influence the developing embryo and program its future health. This incredible discovery is just the beginning of understanding the surprising ways a father's lifestyle can echo through generations. Chapter 2. The MicroRNA Revelation. Having established the remarkable fact that a father's exercise can profoundly influence his offspring's endurance and metabolic health, even without direct genetic inheritance, the pivotal question remains: how is this non-genetic information transmitted? For decades, the focus of inheritance lay almost exclusively on DNA. Paternal non-genetic inheritance was once controversial, and sperm small RNAs were often overlooked. However, cutting-edge research has now revealed the surprising answer. Tiny molecules within sperm, known as microRNAs, serve as the unexpected carriers of these intergenerational exercise benefits. The scientific spotlight then intensified on sperm small RNAs. MicroRNAs, often abbreviated as miRNAs, are a class of small, non-coding RNA molecules, typically only about 22 nucleotides long. They are part of a broader category of small RNAs, defined as those less than 200 nucleotides long. Unlike messenger RNA, which carries instructions for making proteins, microRNAs don't directly code for anything. Instead, they act as powerful regulators, like tiny molecular switches or dimmers, controlling which genes are turned on or off and how much protein is made from a particular gene. They are known to mediate communication within cells and between different tissues, and even across species. The emerging hypothesis was that these small RNAs, influenced by the father's lifestyle and physiological state, could be delivered by sperm during fertilization and then reprogram gene expression in the early embryo, leading to lasting physiological changes in the offspring. To rigorously test this hypothesis and establish a direct causal link, a truly ingenious experiment was devised. Researchers collected small RNAs from the sperm of two groups of fathers, those who had undergone an 8-week exercise training regimen and those who had remained sedentary. These collected small RNAs were then carefully injected into normal, unfertilized mouse zygotes, essentially creating artificial embryos where the only paternal contribution beyond DNA was these specific small RNA molecules. These manipulated zygotes were then allowed to develop and were implanted into surrogate mothers, leading to the birth of offspring. The results were astonishing and provided the most direct evidence yet. Only the offspring that developed from zygotes injected with sperm small RNAs from the exercised fathers reproduced the exercise-trained phenotypes. These injected offspring exhibited enhanced endurance capacity, improved metabolic parameters, and molecular adaptations in their muscles that were strikingly similar to those observed in offspring naturally sired by exercising fathers. In contrast, offspring receiving small RNAs from sedentary fathers or those receiving longer RNA molecules, defined as those greater than 200 nucleotides long, from exercised fathers showed no such benefits. This experiment was a scientific triumph, clearly demonstrating that sperm small RNAs, and specifically microRNAs, are indeed the crucial molecular couriers responsible for transmitting the benefits of paternal exercise across generations. They were literally carrying the biological memory of the father's physical activity, encoded as epigenetic information within these RNA molecules, into the next generation. But how does paternal exercise actually change the microRNA profile within sperm? The research indicates that both regular exercise training and the muscle-specific overexpression of peroxisome proliferator activated receptor gamma coactivator 1 alpha, or PGC1 alpha, actively remodel the landscape of microRNAs within sperm. This means that the father's physiological adaptations to exercise, the increase in mitochondrial function, the shift in muscle fiber types, the overall metabolic efficiency, somehow translate into specific changes in the types and quantities of microRNAs packaged within his sperm. While the exact pathways by which exercise influences sperm microRNA composition are still areas of active research, it's clear that the father's active state creates a unique signature of microRNAs that reflects his improved fitness. The study specifically identified 10 specific microRNAs in mice that were significantly elevated in the sperm of exercised fathers. Intriguingly, similar patterns were observed in human studies, where frequently exercising men showed elevated levels of 7 microRNAs, homologous to those found in the mice, suggesting a conserved mechanism across species. The next critical step was to unravel the molecular mechanism by which these paternally derived microRNAs exert their influence once they enter the egg. The investigation revealed a sophisticated regulatory pathway. The specific microRNAs carried by sperm from exercised fathers directly target and suppress a molecule called nuclear receptor core repressor 1, or NCOR1, within the early embryo. This early embryonic suppression of NCOR1 by paternally inherited microRNAs initiates a reprogramming of the embryo's transcriptional networks, promoting extensive mitochondrial biogenesis and enhancing oxidative metabolism. This lays a fundamental biological groundwork for improved endurance capacity and metabolic health that lasts into adulthood. A more detailed exploration of this intricate molecular pathway will be presented in Chapter 4. The discovery that sperm microRNAs serve as these unexpected carriers of intergenerational exercise benefits is a testament to the intricate and dynamic nature of heredity, extending far beyond the simple transmission of DNA. It opens up a new frontier, urging us to consider how other paternal lifestyle factors might similarly be encoded and transmitted, shaping the health and resilience of future generations. Chapter 3. The Inherited Edge. Offspring Endurance Building. Understanding that a father's exercise habits, mediated by sperm microRNAs, leave an indelible mark on his offspring. This chapter delves deep into the remarkable remodeling that occurs within the next generation's muscles and metabolic systems. We explore what this biological legacy actually looks like, revealing how these offspring are intrinsically primed for enhanced aerobic capacity and robust metabolic health, even before they take their first steps or engage in any physical activity of their own. Imagine a child born with an innate athletic advantage, a natural predisposition for endurance and efficient energy use. This isn't a fantasy, it's the reality observed in the offspring of exercise-trained fathers. When researchers meticulously assess the physical capabilities of these F1 generation offspring, the differences were striking. In demanding incremental treadmill tests where performance is pushed to the limit, these offspring displayed a significant increase in both running duration and the total distance covered. To put a finer point on it, some observations showed them running up to 30% longer and covering nearly 40% more distance than their counterparts born to sedentary fathers. This wasn't a fluke. It was a consistent, profound enhancement of their intrinsic endurance capacity. Beyond simply running longer, the quality of their performance was also notably superior. Following maximal exercise, the offspring of active fathers exhibited significantly lower levels of blood lactate. For those unfamiliar with exercise physiology, lactate accumulation is a key indicator of muscle fatigue. When your muscles are working hard and start to burn, that's often lactate building up, signaling that your body is struggling to clear metabolic byproducts. Lower lactate levels post-exercise signify a superior ability to sustain effort, delay fatigue, and recover more efficiently. This points to muscles that are not only more powerful, but also more resilient. The benefits extended to less strenuous activities as well. In low-intensity sustained running tests, these offspring demonstrated a marked improvement in their ability to maintain performance over time. Even in tests of raw physical strength and endurance, such as the inverted grid test which assesses grip strength, they remained on the grid for significantly longer periods, demonstrating a notable increase in their ability to sustain effort, again accompanied by lower post-test blood lactate. These consistent findings paint a clear picture. The offspring inherited a fundamental enhancement of their physical performance. But the advantages weren't just about how long or how fast they could run. The intergenerational benefits permeated their entire metabolic profile, shaping their body composition and energy regulation from the ground up. These offspring of active fathers showed a more favorable physical makeup, characterized by a lean body mass that was approximately 5% higher and a fat mass that was about 8% lower. They also boasted improved bone mineral density, suggesting a stronger skeletal system. These are not trivial cosmetic differences. They are indicators of systemic metabolic health. To understand how their bodies processed energy, scientists employed indirect calorimetry, a technique that measures oxygen consumption and energy expenditure. The results revealed that these offspring consistently exhibited higher oxygen consumption, often referred to as VO2, and a greater overall energy expenditure compared to those from sedentary fathers. This signifies a more active and efficient metabolism, akin to having a finely tuned engine that burns fuel more effectively and generates more power. Crucially, this heightened metabolic activity wasn't simply due to them moving around more. Their general locomotor activity remained comparable to the control group, confirming that the improvements were intrinsic biological adaptations. To truly grasp the depth of this remodeling, we need to look beyond observable behaviors and delve into the very fabric of their muscles. Skeletal muscle is a dynamic tissue composed of different fiber types, each specialized for distinct functions. There are fast-twitch glycolytic fibers, like type IIB, which are powerful and explosive but fatigue quickly, and slow-twitch oxidative fibers, like type 1 and type IA, which are built for endurance and resistance to fatigue. Endurance exercise is known to induce a beneficial shift towards these oxidative fiber types in the individual performing the exercise. Remarkably, this same fiber type transition was observed in the offspring's muscles, specifically in key muscles like the gastrocnemius, which is crucial for locomotion. The proportion of type 1 and type 2A oxidative fibers increased significantly, while the proportion of type IIB glycolytic fibers decreased, representing a measurable and beneficial shift. This means their muscles were inherently structured to be more endurance-oriented, with a greater capacity for sustained aerobic work. Hand in hand with this fiber-type transition was a profound reprogramming of mitochondrial mass and metabolic genes within the muscle cells. Mitochondria are often called the powerhouses of the cell, responsible for generating the vast majority of cellular energy through oxidative metabolism. An increase in mitochondrial mass means more of these energy-producing factories are present, ready to churn out ATP, the body's energy currency. The early embryonic reprogramming initiated by the paternal microRNAs effectively sets the stage for a heightened activity of pathways that promote mitochondrial biogenesis. It's as if a genetic switch is flipped early in development, directing the developing muscle cells to produce more mitochondria and enhance their capacity for oxidative energy production. Furthermore, the expression of various metabolic genes within these muscles was altered to favor this oxidative, fat-burning metabolism, moving away from reliance on carbohydrates and glycolysis. This comprehensive cellular remodeling underpins the enhanced aerobic capacity observed at the whole body level. This intrinsic biological advantage also provided a powerful shield against metabolic challenges. The offspring of exercising fathers were found to be protected against obesity-induced metabolic dysfunction, specifically exhibiting resilience when faced with a high-fat diet. This is a significant finding in our modern world, where sedentary lifestyles contribute to a global epidemic of obesity and related chronic diseases. Not only did these offspring maintain a healthier body composition, but they also exhibited enhanced glucose homeostasis, meaning their bodies were better at regulating blood sugar levels. This was achieved in part by promoting more efficient glucose uptake in skeletal muscle, a crucial step in preventing conditions like type 2 diabetes. Their cells were more sensitive to insulin, the hormone that helps glucose enter cells, making their metabolic systems more resilient and less prone to dysfunction when faced with dietary stress. It's important to note the specificity of these transmitted benefits. While fathers who engage in endurance training often experience physiological adaptations in their cardiovascular system, such as an increase in the size of the heart's left ventricle, these specific cardiac remodeling effects were not observed in their offspring. This selectivity highlights that the intergenerational transmission isn't a broad, undirected transfer of all paternal traits. Instead, it appears to be a targeted process, precisely influencing the molecular and cellular machinery that governs skeletal muscle function and overall metabolic efficiency, thereby directly enhancing aerobic capacity. The implications of these findings are profound. They suggest that a father's lifestyle choices, particularly his commitment to exercise, can literally sculpt the physiological landscape of his children, granting them a biological head start in endurance and metabolic health. This is a powerful non-genetic form of inheritance that primes the offspring's body to thrive, offering an intrinsic resilience against the very metabolic challenges that plague modern society. It opens up exciting avenues for understanding how our ancestors' physically demanding lives might have conferred similar advantages, and how we, in turn, can leverage this knowledge to optimize health for future generations. The question now becomes, what are the precise molecular pathways and intricate networks that orchestrate this remarkable transmission and remodeling, allowing a father's sweat to become his child's strength? Chapter 4. The Molecular Choreography of Inheritance. We have now journeyed through the remarkable discovery that a father's exercise can confer significant endurance and metabolic advantages to his offspring. And we've identified sperm microRNAs as the unexpected messengers of this intergenerational communication. The question that naturally follows, and indeed the core of this chapter, is how? How do these tiny molecules orchestrate such profound biological remodeling? What are the precise molecular pathways and intricate networks that translate a father's physical activity into his child's intrinsic athletic and metabolic resilience? The answer lies in a sophisticated, multi-step molecular cascade that begins in the father's muscles, travels via sperm, and culminates in the reprogramming of the early embryo. At the very heart of the father's physiological response to exercise is a crucial protein known as peroxisome proliferator activated platter gamma coactivator 1-alpha, or PGC1-alpha. Think of PGC1-alpha as a master switch or a central conductor for the body's energy systems, particularly within skeletal muscle. When a father engages in endurance exercise, his muscles respond by significantly increasing the production of PGC1-alpha. This upregulation is not arbitrary. It's a direct molecular command that initiates a cascade of beneficial adaptations. PGC1-alpha's primary role is to coordinate the expression of hundreds of genes involved in mitochondrial function and oxidative metabolism. Mitochondria, as we know, are the cellular powerhouses, and PGC1-alpha essentially tells the cell to build more of them, make them more efficient, and switch the muscle's fuel preference towards burning fats rather than solely relying on carbohydrates. It also drives the transformation of muscle fibers from quick-burst, easily fatigued types to slow-twitch, endurance-oriented types. This makes the muscle more resistant to fatigue and capable of sustained effort. To confirm PGC1-alpha's pivotal role in initiating this intergenerational effect, scientists conducted an elegant experiment using transgenic mice. These special male mice were engineered to constitutively overexpress PGC1-alpha specifically in their skeletal muscles, meaning they naturally had high levels of this protein even without exercising. Remarkably, these PGC1-alpha overexpressing fathers, despite being sedentary, displayed endurance-like adaptations, similar to regularly exercised animals. And critically, when these transgenic fathers sired offspring, those progeny exhibited improved endurance capacity and metabolic traits. This observation was particularly powerful because the offspring did not inherit the PGC1 alpha transgene itself. This strongly suggested that it wasn't the direct inheritance of the gene, but rather the physiological state induced by PGC1 alpha in the father's muscles that was somehow leaving an epigenetic mark on the next generation. This established PGC1 alpha in the father as the critical upstream initiator of the entire process. The next crucial step in this molecular relay race is the remodeling of the sperm microRNA profile. The father's exercise and the subsequent surge in PGC1 alpha activity in his muscles doesn't just affect his own body, it profoundly alters the cargo carried within his sperm. In response to exercise training, the specific composition and abundance of microRNAs within the sperm undergo a significant shift. Researchers identified a distinct signature of microRNAs that were either upregulated or downregulated in the sperm of exercised fathers compared to their sedentary counterparts. For instance, several specific microRNAs were found to be significantly elevated in the sperm of physically active individuals in animal models. While the precise mechanisms by which exercise influences sperm microRNA composition are still areas of active investigation, current hypotheses suggest roles for small extracellular vesicles transporting microRNAs to the testes, or direct hormonal responses altering microRNA expression. These microRNAs are essentially packaged with the sperm, ready for delivery at the moment of fertilization. Once fertilization occurs, these paternally derived microRNAs embark on their critical mission within the early embryo. They are not merely passive passengers, they are active molecular agents. The research precisely decoded their immediate target, a molecule known as nuclear receptor copressor 1, or NCOR1. NCOR1 is a protein that acts as a functional antagonist of PGC1 alpha. To reiterate our earlier analogy, if PGC1 alpha is the accelerator for mitochondrial biogenesis and oxidative metabolism, NCOR1 is the brake. It actively binds to and represses the activity of PGC1 alpha, thereby dampening the very processes that enhance endurance and metabolic efficiency. Here's where the ingenious mechanism truly unfolds. The specific microRNAs delivered by the exercised father's sperm directly suppress NCOR1 in the nascent embryo. When these microRNAs enter the egg, they don't immediately alter the embryo's DNA. Instead, they interact with the messenger RNA, mRNA molecules that carry the instructions for making the NCOR1 protein. By binding to these NCOR1 mRNA molecules, the paternal microRNAs prevent them from being translated into functional NCOR1 protein. This precise act of gene regulation, effectively reducing the amount of NCOR available in the early embryonic cells, was powerfully demonstrated when scientists showed that injecting even a single specific microRNA from exercised fathers into normal zygotes was sufficient to reproduce the endurance benefits in offspring. The consequence of this NCOR1 suppression is profound. It effectively takes the foot off the brake for PGC1 alpha activity in the developing embryo. With its antagonist diminished, the embryo's own PGC1 alpha is unleashed, free to exert its full influence. This leads to a fundamental reprogramming of the embryo's transcriptional networks. From these earliest stages of life, the cells are directed to activate genes that promote mitochondrial biogenesis and oxidative metabolism. This isn't just a minor tweak, it's a foundational resetting of the cellular machinery responsible for energy production and utilization. The embryo, from its very inception, is being programmed to build more efficient powerhouses and to favor an endurance-oriented metabolic profile. By resetting the gene regulatory networks that govern energy metabolism and muscle development, the paternal microRNAs sculpt the offspring's physiology from the ground up. This precise molecular pathway involving paternal PGC1-alpha, the exercise-induced remodeling of sperm microRNAs, and the subsequent suppression of embryonic NCOR1 forms a coherent intergenerational regulatory axis. This axis causally transmits the exercise-induced phenotypes and metabolic adaptations, laying the groundwork for improved endurance capacity, enhanced glucose homeostasis, and a more favorable body composition that persists throughout the offspring's adult life, even without direct exercise training. This intricate dance of molecules not only illuminates the causal role of each component, paternal PGC1-alpha as the initial sensor and amplifier of exercise signals, sperm microRNAs as direct epigenetic carriers, and embryonic NCOR1 as the critical molecular switch, but also opens exciting avenues for future research. This detailed understanding of the molecular choreography will be crucial for further investigating how these epigenetic mechanisms are finely tuned, how the microRNA profile in sperm is precisely remodeled by exercise, and how these pathways might be influenced by other lifestyle factors, ultimately allowing us to fully harness this intergenerational legacy. Chapter 5. The Echo of Fitness, Shaping Future Health. Our journey through the intricate world of paternal exercise and its surprising legacy has revealed a truly revolutionary understanding of inheritance. We've uncovered the molecular symphony orchestrated by sperm microRNAs, translating a father's active lifestyle into an intrinsic biological advantage for his offspring. Now it's time to step back and consider the profound implications of these discoveries, not just for scientific inquiry, but for our collective approach to health, family well-being, and even the future trajectory of human generations. This is really a novel paradigm, noted epigeneticist Colin Canine, underscoring the revolutionary nature of this research. One of the most significant shifts it brings is an expanded appreciation for the functional repertoire of microRNAs themselves. This groundbreaking work elevates sperm microRNAs to an entirely new role, that of facilitators of intergenerational communication, revealing them as mobile signaling molecules capable of transcending the boundaries of a single generation, as detailed in Chapter 2. This means that a father's dynamic experiences, such as his commitment to physical activity, can be encoded as epigenetic information within these sperm microRNAs, influencing the biological blueprint of his children. Intriguingly, similar patterns were observed in human studies, where frequently exercising men showed elevated levels of 7 microRNAs homologous to those found in the mice, reinforcing the human relevance of this discovery. Explain with examples, but do not use analogies that are not truthful. This understanding initiates what has been aptly described as a butterfly effect on embryonic development. The subtle yet precise intervention of paternally inherited microRNAs in the early embryo, as explored in Chapter 4, lays a foundational groundwork for enhanced endurance capacity and improved metabolic health, establishing a trajectory that persists into adulthood. This concept challenges the long-held view that the embryo is a blank slate, primarily shaped by maternal influences and its own genetic code. Instead, it underscores a powerful paternal contribution that actively participates in the very initial stages of developmental programming. The implications for public health are nothing short of transformative, especially in an era grappling with the global challenges of physical inactivity, sedentary lifestyles, and the alarming rise of obesity and chronic metabolic diseases. As the Trends Endocrinology Medeb article succinctly put it, "Run for your sons, paternal exercise boosts offspring fitness." This research offers a rational and remarkably cost-effective strategy to improve the health of future generations, particularly when compared to expensive pharmacological interventions or complex medical treatments. By demonstrating that paternal exercise, even prior to conception, can lead to significantly enhanced glucose homeostasis and more efficient glucose uptake in skeletal muscle in offspring, as detailed in Chapter 3, it provides a tangible pathway to combat the intergenerational cycle of metabolic dysfunction. Imagine a world where lifestyle interventions, specifically a father's commitment to regular physical activity, could proactively confer a degree of resilience against conditions like type 2 diabetes and obesity to his children. This isn't a distant dream. It's a scientific possibility emerging from these findings. It suggests that promoting paternal well-being isn't just about the father. It's an investment in the health of his entire lineage, offering a natural and accessible form of preventative medicine. This new perspective also demands a critical reevaluation of our conventional focus in reproductive health. For decades, the spotlight has predominantly been on maternal health during pregnancy, and rightly so, given the profound and direct influence of the mother's physiological state on fetal development. However, this research firmly establishes paternal behavior as a critical and previously underestimated determinant of offspring well-being. It highlights that the health of the father, particularly his lifestyle choices in the preconception period, holds substantial sway over the physiological and metabolic characteristics of his children. This doesn't diminish the mother's role, but rather expands our understanding to encompass a truly bilateral contribution to inherited health. This expanded view could, and perhaps should, lead to the development of new guidelines for preconception paternal lifestyle modifications, emphasizing that both parents contribute significantly to the health blueprint of their future children. Looking ahead, this pioneering work opens up thrilling new frontiers for scientific exploration and the potential for innovative health interventions. While we have decoded a precise molecular pathway involving paternal PGC1-alpha, sperm microRNAs, and embryonic NCOR1, the full spectrum of microRNAs involved in transmitting exercise benefits is likely much broader. Researchers are now eager to explore whether other microRNAs, acting in concert or independently, contribute to the diverse physiological adaptations observed in offspring. Furthermore, the interplay between these microRNAs and other epigenetic mechanisms, such as DNA methylation and histone modifications, remains a rich area for investigation. It's possible that sperm microRNAs might not only directly regulate gene expression, but also influence or interact with these other epigenetic marks, creating a more complex and robust system of intergenerational information transfer. Beyond exercise, this discovery invites us to consider how other paternal lifestyle factors might similarly sculpt offspring phenotypes. Could a father's diet, his stress levels, or even his exposure to certain environmental toxins be encoded within sperm microRNAs, translating into specific health advantages or vulnerabilities for his children? This concept of biological memory, encoded not just within the static sequences of DNA, but within the dynamic and responsive molecules of RNA, is a paradigm shift. As one source vividly put it, within a sperm's minuscule head are stowaway molecules, which enter the egg and convey information about the father's fitness, such as diet, exercise habits, and stress levels, to his offspring. It suggests that our life experiences, far from being confined to our individual bodies, can literally echo through the generations, leaving an imprint on the metabolic destiny of our descendants. Understanding these connections could inform precision health strategies, allowing us to tailor interventions based on an individual's paternal history and current lifestyle. However, it is crucial to acknowledge the nuances and limitations that guide future research. For instance, some observations hint at potential sex-specific differences in how paternal exercise benefits are manifested, with male offspring sometimes exhibiting more pronounced advantages than females. Unraveling the reasons behind these potential differences will be vital for developing truly equitable and targeted interventions. Moreover, while the benefits observed are profound, current evidence suggests they are primarily transgenerational, meaning they primarily impact the immediate next generation, the F1 offspring, rather than being heritable across multiple generations without further intervention. This implies that the beneficial reset requires continuous input from each generation's healthy lifestyle choices, reinforcing the importance of ongoing physical activity rather than a one-time fix. Another key area for future inquiry concerns the precise mechanisms by which exercise actually changes the microRNA profile within sperm. Is it a direct hormonal response to physical activity that alters microRNA expression in the testes? Or do other signaling molecules, perhaps small extracellular vesicles released from active muscles, act as carriers, transporting specific microRNAs to the developing sperm? These are complex biological questions that demand further investigation. Similarly, while the focus has been on muscle and metabolic adaptations, exercise has myriad benefits for other organ systems, including the cardiovascular system, liver, and adipose tissue. Future research will undoubtedly explore whether paternal exercise through these microRNA pathways also influences the function and health of these non-muscle tissues in offspring. Ultimately, these revelations are more than just scientific breakthroughs. They are a profound invitation to consider the far-reaching legacy of our daily habits. They emphasize how the benefits of physical activity transcend the individual, echoing through the lives of future generations in the subtle yet powerful language of microRNAs. By bridging exercise physiology, molecular biology, and epigenetics, this pioneering work charts an inspirational course toward unlocking the secrets of intergenerational health transmission. It challenges us to consider our own lifestyle choices not just as personal decisions, but as contributions to a broader biological heritage, shaping the very resilience and potential of those who will come after us. This knowledge empowers us to look beyond immediate gratification and embrace a vision of well-being that spans across time, offering hope for a healthier, more enduring future. Our journey has revealed a groundbreaking truth. A father's exercise sculpts his offspring's endurance and metabolic health through sperm microRNAs. This epigenetic legacy, initiated by paternal PGC1 alpha and transmitted via microRNAs suppressing embryonic NCOR1, fundamentally reprograms the child's biology for enhanced fitness and resilience against metabolic disease. This paradigm-shifting discovery expands our understanding of inheritance beyond DNA, highlighting the profound impact of paternal lifestyle. It offers a powerful, cost-effective strategy for public health, urging us to reevaluate preconception health and consider a father's active choices as an investment in his lineage. The echo of our fitness truly shapes the future. Thank you for listening.